Welcome to this lecture on molecular hormone action. Today we are going to explore the fascinating communication system of the human body. We will learn exactly how chemical messengers travel through the bloodstream and instruct specific cells to change their behavior, keeping us alive, balanced, and healthy. Let us dive in. To begin, imagine you are standing in a crowded room where everyone is shouting different messages at the same time. If you are waiting for a specific message from a friend, you will only react when you hear your name or a secret code word. In the human body, hormones are like those shouted messages, traveling through the bloodstream, reaching every single cell. However, a cell will only hear and respond to the hormone if it has the right ear, which we call a receptor. Now that we understand what a receptor is, let us look at what happens when a hormone actually finds one. Molecular hormone action refers to the specific biochemical mechanisms by which a hormone molecule binds to its target cellular receptor. This binding initiates a cascade of intracellular events. This process, often called signal transduction, bridges the gap between an extracellular signal on the outside and an intracellular physiological response on the inside. It is the fascinating story of how a chemical message is received by the cell and translated into a physical action, like making a protein or releasing sugar. Building on this idea of signal transduction, we must recognize that not all hormones deliver their messages in the same way. Depending on the chemical nature of the hormone, this action occurs either at the cell membrane or deep inside the cell. For water-soluble hormones, like insulin, the receptor is on the surface, and binding triggers secondary messengers inside the cell. For lipid-soluble hormones, like testosterone, the hormone actually enters the cell, binds to a nuclear receptor, and directly alters gene transcription. Ultimately, it is a precise sequence of molecular dominoes falling to change the cell's behavior. Understanding these molecular dominoes is not just academic, it is absolutely critical for medicine. This knowledge explains how our bodies maintain balance, grow, and respond to stress. In the real world, it is the foundation of modern pharmacology. For example, when a patient has type 2 diabetes, their body produces insulin, but the molecular action at the receptor level is broken, leading to dangerously high blood sugar. By understanding these exact molecular pathways, scientists can design life-saving drugs like beta blockers for heart disease or synthetic thyroid hormones for hypothyroidism. To truly master how hormones act on a cell, we must move from biology to mathematics. The relationship between the concentration of a hormone and the number of receptors it occupies is described by the fractional occupancy equation. In this formula, Y represents the fractional occupancy, which is the percentage of total receptors currently bound by the hormone. The symbol H in brackets stands for the concentration of the free hormone in the blood. Finally, Kd represents the dissociation constant, which measures the receptor's affinity. A lower Kd means the receptor binds the hormone very tightly. When hormone concentration increases, occupancy increases, and the cellular response grows stronger. Now that we have our formula, let us apply it to a real clinical scenario. Let us imagine a patient whose pancreatic beta cells are releasing insulin. The concentration of insulin in the blood is 10 nanomolar, and the dissociation constant for the insulin receptor is 2 nanomolar. We want to find the fractional occupancy. We substitute H with 10 and Kd with 2 in our equation. Simplifying the denominator by adding 2 and 10 gives us 12. Dividing 10 by 12 gives us 0.833. This means 83.3% of the receptors are bound, sending a very strong signal to the muscle cell to take up glucose after a large meal. Having seen how to calculate occupancy from a known concentration, we can now look at how pharmacologists use this in reverse. When designing drug dosages, a doctor often wants to achieve a very specific target occupancy to balance a patient's metabolic rate. By setting a target for Y, we can reverse engineer the equation to find the exact hormone concentration required in the blood. Let us explore a scenario involving a synthetic thyroid hormone, where the goal is to achieve exactly a 50%, or half maximal, receptor activation. Let us mathematically derive the concentration needed for this half maximal response. A doctor wants exactly 50% of the patient's thyroid receptors occupied, meaning Y equals 0.5. The dissociation constant of this synthetic hormone is 5 nanomolar. We plug in 0.5 for Y and 5 for Kd. Our goal is to isolate the unknown hormone concentration, H. To remove the fraction, we multiply both sides of the equation by the denominator, bringing the terms up to the same level and making the algebra much easier to handle. Continuing with our derivation, we now distribute the 0.5 into the parentheses on the left side. Half of 5 gives us 2.5, and half of H gives us 0.5H. This expands the left side so we can start moving terms around. Next, we subtract 0.5H from both sides to group all the H terms together on the right side of the equation. Since we had one full H on the right, subtracting half of an H leaves us with exactly 0.5H. We are getting very close to isolating our target variable. In our final step, we divide 2.5 by 0.5, which gives us a required hormone concentration of 5 nanomolar. Notice something fascinating here. The required concentration of 5 nanomolar is exactly equal to the dissociation constant of 5 nanomolar. This beautifully illustrates a fundamental rule of molecular endocrinology. When the hormone concentration exactly equals the dissociation constant, exactly half of the receptors are occupied. Understanding this mathematical relationship allows medical professionals to treat patients with incredible precision. Let us visualize everything we have learned. On the top of this diagram, you can see the physical process of signal transduction. A hormone binds to a surface receptor, specifically a G protein-coupled receptor, which then activates adenylate cyclase to produce secondary messengers inside the cell. On the bottom, we see the mathematical reality of this process, plotted on a graph. The horizontal axis represents hormone concentration, and the vertical axis represents fractional occupancy. As concentration increases, the curve rises steeply, but eventually levels off as it approaches a maximum occupancy of 1. This shows that once all receptors are bound, adding more hormone will not increase the cellular response. To summarize our lecture, molecular hormone action is the fundamental process by which chemical messengers instruct cells to change their behavior. We learned that hormones rely on specific receptors, either on the cell surface or inside the nucleus, to translate their presence into a biological response. By using the fractional occupancy equation, we can precisely predict these responses. And we proved that when hormone concentration equals the dissociation constant, exactly half the receptors are occupied. This beautiful lock-and-key mechanism is the very foundation of how our entire endocrine system maintains harmony and health. Thank you for joining me in this exploration of endocrinology.